Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have been discussing over the last few classes the algorithms that are used for read mapping. Okay, so, we have discussed why this is a very challenging problem. We have a huge number of reads, we have a big genome and we need to find the location of those reads in the reference genome, right. So, this process it kind of combining all these factors, it starts to take a lot of time, right. So, that is why we need specialized algorithms and we have discussed two such algorithms so far. We have talked about hash table based algorithms and we have in the last class talked about suffix tree and suffix array based mapping algorithms. So, in this class we will be talking about Burrows-Wheeler transform based algorithms. So, Burrows-Wheeler transform is a specific transform of the string or the reference sequence and we use that transform to develop mapping algorithms and we map reads using that transform. Okay, so, this is what we will be discussing in this class Burrows-Wheeler transform based mapping. We will discuss what this Burrows-Wheeler transform is and then we will discuss the mapping algorithm. So, these are the keywords that will come across in this presentation. One is string rotation, another is lexicographical sorting and the third is left mapping. So, we will discuss these concepts in detail, but you will see these terms throughout the presentation. So, just to briefly recap the algorithms, the mapping methods that we have discussed so far. Okay. So, we have talked about hash table based mapping methods, we have talked about suffix tree based methods and suffix array based methods, right. So, hash table based method, right. So, we talked about how we stored the reference sequence in a hash table, right. We generate seeds and we store in hash table along with the locations and we generate similar seeds from the read data and we search in the hash table and find the locations. Okay. So, of course, there are a lot more details in there. And then we talked about suffix tree and suffix array based methods. These are related ideas. So, they both relate to suffixes, right, and we talked about how we generate suffixes from a reference sequence and then we talked about the tree representation and the array representation, right, and we talked about these advantages, disadvantages and how we can search sequences using the suffix tree or suffix array structure. Okay. And to summarize, right, we have seen hash table and suffix tree based methods are really fast, right. So, we can map it very quickly and if you compare with BLAST or BLAT or brute force even, right, these are incredibly fast. Okay. But the problem is they require large amount of memory or storage, right. So, all these methods they take at least 12 to 15 GB of RAM if you are working with human genome, right, which means you probably would not be able to run this in the desktop system, right. Suffix tree for example, it takes even more, right, it takes about 30 to 35 GB RAM if you are working with human genome, right. So, because of that elaborate data structure. So, the other disadvantage is that this building these structures, data structures, right, this take quite a bit of time, right. So, you have to build this hash table, means you have to extract those seeds, right, and then represent them as keys and then stored also the values, right, all that takes time. Similarly, building the suffix tree structure, right, that also takes quite a bit of time, right. So, the time is there that you have to give for at least generating this data structure. In case of suffix tree or array, right, once you generate this data structure you can store it, right, you do not have to build this again and again, whereas for hash table if you change the seed size, the k length, right, you will have to rebuild the hash table. Again the k length can change depending on the sequencing platform that you are using, the read length that you are using and the sequencing error that you expect in the data. What we have also seen is that hash table based methods are poor at mapping reads to repeat regions, right. So, you have it will take more time because you have to do this chain and alignment or chain extend, right, for different positions, right, multiple positions and that increases time. On the other hand, suffix tree and suffix array based methods can map reads to repeat regions very quickly, right. So, we have this because of the data structure this is possible, but these are very poor at mapping reads with mutations or sequencing errors, right. So, this is something we have seen because they will not be able to find matches in the data structure. So, they will return, right, there is no match. So, this is not suitable, right. If you are discarding a read because there is a mismatch, then we will probably discard a large fraction of the data. So, is there a way to do better, right. So, what we have seen these two algorithms, they can do some of these tasks pretty well, but we probably need a more efficient algorithm, right. So, what we need actually, this is kind of like the wish list, right. So, we have we need an algorithm that does not take up too much memory or storage, right, like the earlier ones. Okay. So, we can run it in our computer, in the desktop, or in the laptop even, right. It should be fast and scalable. So, if you are dealing with billions of data, it should not be a problem, right. Should be able to complete the mapping within a reasonable amount of time. And then of course, this method should be able to handle reads with mutations and sequencing errors, because this is part and parcel of next generation sequencing data, right. So, you will have reads with sequencing errors and mutations, and also it should be able to map reads to repeat regions quickly, right. It should not be taking too much time to do this process. Okay. So, can we design this kind of algorithm, or is there an algorithm that can do that, right. So, what researchers looked at is something called Burrows-Wheeler transform. Okay. So, this is a transform applied on strings, and this idea was taken from there and applied to read mapping algorithm. So, what we will discuss now is this Burrows-Wheeler transform based mapping methods. So, first we will talk about Burrows-Wheeler transformation, or in short BWT. Okay. So, what are the steps? Okay. I will first describe the steps, and of course, we will take examples and we will implement this and see how this actually works. Okay. So, the step is first we add a dollar sign to the end of the string. So, if you are taking a reference genome or reference sequence, we add this dollar to the end. Okay. And with the assumption is that dollar being lexicographically smaller than all letters of the alphabet, right. So, especially if you have ATGC etc., dollar is smaller than that. So, when you do sorting, right, dollar will come first. Okay. So, it kind of hints at like there would probably be a sorting step later on, right. Then what we do is something called string rotations. Okay. So, we generate all possible string rotations from the reference sequence. Okay. So, we will see how this works in a moment with an example. Once we have generated these string rotations of the original reference sequence, right, what we do is something called sorting. Okay. So, we sort the original string and its rotations lexicographically. Okay. So, we have seen lexicographical sorting just in case of suffix array. So, here is a similar idea, right. We generate all these rotations and take the original string and sort lexicographically. And this matrix that we generate, the last column of that matrix is the Burrows-Wheeler transform of the original string. Okay. And then this matrix, this Burrows-Wheeler transform has certain properties that you can utilize for read mapping. Okay. So, we will slowly elaborate this whole process starting with the Burrows-Wheeler transformation. Okay. So, let us take an example. Okay. So, here is the reference sequence we have taken. Okay. So, we have this basis. So, the step 1 is add dollar, right. So, that is what we have done here, right. We have added this dollar at the end. Okay. The next step is the rotation of the strings. Okay. So, the first rotation, right, we are rotating, imagine you are rotating in clockwise direction, right, clockwise or anti-clockwise, right. So, in one direction, right. So, how do you rotate the string, how do you visualize this process? So, imagine you are right, you have written these letters on a cylinder, right. So, you have a pointer at a specific position in the cylinder and you read the letter in that position, right, and that is the start of the string, and rest of the letter are around this cylinder, right, and you are rotating this cylinder and you are just visualizing the start position. Of course, the start string start letter will change, right, and the string will rotate. So, this is the process, that is what we are doing. Okay. So, you can visualize this, we are bringing dollar first and then we are shifting the rest towards the end, right. So, then we have this dollar first, then we have GSCG, TSC, GTCA, right, and we continue this process until we reach go back to the original string. Okay. So, let us do all these rotations carefully, have a look, right, what we are actually doing. So, what is the next step then? So, we will bring this last A to the first position, right, and then shift rest of them to the end. Okay. So, this is the process actually, and you can carefully see, right. So, what we have done is we have done these rotations. Okay. So, here you will see actually we have actually kind of taken the dollar first, right. This is first here, yeah, and then we have got this A here from to the beginning, and then we have pushed rest of the them towards the end. Then we have put this A here, right. So, we have now have two A's in the beginning, and we have pushed the rest towards the end, right. So, this process continues, right. So, then we get the C, this C comes here, then we get the T here, right, and so on, right, and we continue until we reach this original string, right. So, this is the last string, right, because if you put G in the front, what we will get is this one, right, we will get back to the original string. Okay. So, we continue this rotation and we generate all possible rotations. Okay. So, this is the step 2, and this gives us the whole matrix. Okay. So, this is the matrix that will generate through this string rotation. Okay. So, once we have generated these rotations, we take the original string and the rotations and we do the sorting. Okay. Here we go back to the sorting now, sorting step, right. So, please have a look carefully, right, which one would be the first one, right, which would be the first here, right. Once we sort all these strings, which one would be the first string to that we pick. Okay. So, the first one would be this one, right. Remember we mentioned dollar is smaller than the rest of the letters, right. So, this will come first. Okay. So, what we do, this will be the first. So, we bring it to the first position. Okay. So, this is the first one, first string. Then from the rest we have to choose the second one now. Okay. So, the second one is again so it should start with A, I think, right. So, we have 4 letters A, T, G, C. So, if we sort that lexicographically, we have A, C, G and T. So, all the strings starting with A will come now. Okay. Now, out of those, the strings that are starting with A, right, you have 1, 2, 3, 4, which one would come first? Okay. So, if the first letter is same, we look at the second letter. Okay. And if we look at the second letter, we see this one has dollar, right. Again, remember dollar is smaller than all the letters. So, this one should come now. Okay. So, this is the one that should be now taken up, right. So, we will move this here, we will take this here after the so this will form the second string, right. So, this is the first, this is the second, right. So, this is the sorted part, right. First two are now sorted. Now, we have to sort with the rest of the matrix. Okay. So, for the third one, for the third position, what do we have? You have again these ones with A's, right, and first letter is same for all of them, A is the first letter, but for this one second letter is A, for this one second letter is C, second letter is C here, right. So, surely this one will now has to be moved here, right. So, this will move. Okay. So, this one is we have chosen and this will move here, all right. So, this is the third one. Now, this is the sorted part, right. So, we continue, right, and for this one we have now this one here. So, again we have two strings that start with A, and the second letter is also same for these two, right, C, C. Third letter is also same, right, fourth letter is also same. Okay. So, we keep on, right, if they are same, right, keep on going, right, C, G, G, T, T, and fifth letter we have now resolved this, right. So, fifth letter, this one has A, the other one has C. Okay. So, this one, the one with A will come, right. So, this will be now brought up here. So, this will come here, right. So, this part is now sorted. Okay. And we have to continue this process. We have one more with A. So, this one will surely come up now, right. This will if you sort this, right, this one will come up here. Okay. So, this is getting sorted slowly. Now, we have exhausted all the strings that start with A, right. So, we need to look at now strings that start with C, right. Again there are 3, right, that start with C. Then we look at the second letter, we have A, we have G, we have G. Okay. So, this one starts with A, the second letter is A, right. So, we have will choose this one, okay, and this will come up here, okay, and we have sorted this one. We have 2 more with C's, right, and again if you do the same comparison, you will see this one is A in the 4th position and this one has C, right. So, this one will get priority, of course, right, the one with A, and this will be brought here, okay. So, this is the sorted part now, and we have just one more remaining with C. So, this will come up, right, and we have generated the sort. Now, out of these 5 that are remaining, we have strings that start with G and some strings start with T, right. Again the G will get preference, right, and again we have very similar order, right. So, you see G and A, G T, G T, right. So, here you have G T A, here you have G T C. So, you can now guess the order, right. So, this one will come first, then this one, then this one, right. So, we can actually go through this quickly, right. So, this one will be first, this is then this one, right, is taken up, and this one here, right. So, this is now sorted, and then finally these 2, right. So, you have TCA and TAC, it is easy, right. So, this one will get preference, you have A in the second position, right. So, this will come up, and this is the final one, and we have generated the sorted list. Okay. So, this is the sorted matrix that we have for all the strings and that we have generated by rotations. Okay. So, in this matrix now we will perform all sorts of tricks, and we will see how this can help us map those reads. All right. So, once we have done this process, we have done these 3 steps. Okay. This is the Burrows-Wheeler transformation of the original string. Okay. So, through these steps, right, we have generated this Burrows-Wheeler transformation, and we simply write this, right. So, this is the original string or original reference sequence, and we have generated the Burrows-Wheeler transformation, which is the last column of this matrix. Okay. So, what you see, what you probably notice is that this structure, this transform actually generates a string that is of same size as the reference sequence, right. So, it is just +1. So, you have a dollar here extra, but this is roughly the same size as the reference sequence of the reference genome, right. So, which means it would probably not take too much space in the memory, right. So, that is a very important consideration for us, right. So, earlier methods that we have talked about, they all took a lot of memory, right, lot of memory and we could not run them in desktop computer for that reason, right. Here it seems the memory will be memory requirement will be less, and perhaps we will be able to run this in desktop computer. We will see in a moment. Okay. So, this is the Burrows-Wheeler transformation of the original string, and we will utilize this information to actually do the mapping. Okay. So, it turns out there are certain properties of this transform. Okay. So, BWT actually has certain properties that we can utilize to do read mapping. Okay. So, we can also add positions, we can keep track of these positions of each of these letters, right, and we can store these positions. Again, this is this will not take too much probably memory or storage space if we have to store these positions. Okay. All right. So, what we have discussed, right, BWT size is the length of the reference, right. Okay. Now, what is stored actually in the memory? So, we can actually simply store this BWT, right, the last column, and it turns out you can actually generate the first column of that matrix from this BWT information alone. Okay. So, if you just simply store this information, if you store this column in the memory or in a file, right, because once you have generated this BWT for a reference sequence or reference genome, you can store it, right, and every time you have to do mapping, you can simply load that information into memory. Okay. And it turns out if you store this BWT, you can actually generate this first column, okay, because the first column is lexicographically sorted, right. Just by sorting this BWT, you can generate the first column, and you can probably match this against the matrix. Okay. So, this information is very helpful, because if you just store this information here along with positions in a file, when you are running this program, you can generate the first column. Now, why do we need the first column? Okay. So, the BWT, remember, is the last column, and this is the first column of this matrix, and it turns out there is a property called last first mapping or LF mapping. Okay. So, this property of BWT is very useful, and we can utilize this property to actually do do the red mapping. Okay. So, what do I mean by this last first mapping? Okay. So, here is the first column I have written by F, and the last column is written by L, which is also the Burrows-Wheeler transform or BWT. So, it says last first mapping says is that the ith occurrence of character C in the last column and the ith occurrence of character C in the first column, they both correspond to the same character in the reference sequence. Okay. So, they are actually the same character in the reference sequence. If you count, let us say, fourth occurrence of character, let us say the base A in the last column and fourth occurrence of the character base A in the first column, they correspond to the same base in the reference sequence. Okay. So, what it means now is, if we let us say number these A's in the first column by A1, A2, A3, A4, right, we are distinguishing these 4 A's by these numbers or these subscripts. Okay. What it means is that these 4 A's, right, they actually correspond to these 4 A's here, okay, in the last column. Okay. So, this correspondence is shown by these red arrows. Okay. And if that is the case, if they are corresponding to the same base in the reference sequence, we can also number these A's by A1, A2, A3 and A4. Okay. So, this is what we mean by last first mapping. Okay. So, this order of appearance of these letters, right, they remain same in the first column as well as in the last column. Okay. And we can now extend this to all bases, right, and we can number these occurrences of C, G and T by these numbers, the subscripts C1, C2, C3, G1, G2, G3 and T1, T2, right, and they will again correspond to the bases or the order of occurrence in the last column. Okay. So, this is the last first mapping. Okay. So, this is the property that is very helpful for read mapping. Okay. So, we will see this, right, how we can actually utilize this information to actually do read mapping. Now, we will talk about another property, right, what we can do with LF mapping. Okay. So, using this BWT, right, sequence and the property of LF mapping, we can actually generate the original reference sequence. Okay. Only thing we need is this BWT, and we can generate the first column from BWT, and combining these two with the principle of LF mapping, we can go back to the reference sequence. Okay. So, I will illustrate that in a moment, right. So, here is the first column, right, and we have the last column, which is the BWT. Okay. And remember, we can generate first column from BWT. We have generated this, and what we will do is we will do this last first mapping or something called traversing, right. So, we will start with this first letter in the last column, okay, and we will see, right, we will map this to the corresponding A, right. So, this A1, it corresponds to this one here, right, because of the LF mapping property. Okay. Now, this A1, what is the letter that is before this A, right. Remember, the string rotation we are doing in such a way, right. So, we are bringing these letters, maybe you can have a look, right, at the matrix, and that will make things clear, right. So, when we are doing the string rotation, let us say here, what it means is that if you look at this string, this is the letter that is before this A, right. This A is before this A here in the reference sequence. Similarly, if you take this example for this string, this T occurs just before this C in the reference sequence. Okay. So, the string rotation actually means this, right. So, we can actually map the letter that is before this letter in the first column. Okay. So, this is something what we are actually getting here, right. So, let us go back there, okay, and here we are, this A1 corresponds to this one. Now, because of this property, because of the string rotation, what it means is that this A, right, occurs before this A, right. So, A1 and A2 occurs before A1 in the reference sequence, right. So, we have now written this reference sequence, we are writing this reference sequence, right. So, we have A, right. So, we have written down 2 A's. Okay. Now, similarly, so this C occurs before this A, right. So, we can do this mapping again, right. So, what it means, we have now added C, right, because this C occurs before this A, okay. And then this C, for this C T, this T is mapped, right. This T occurs before this C, we have added this T now in for that in the reference sequence that we are building, right. And for this T, we can see this G will be before this T, right. And we have added G now to the reference sequence, and then we again use the last first mapping here, right. So, this G3 corresponds to this G3 here, right, and we can continue this process. We can see this C is before this G because of the rotation. Again, we go back here to the first column using last first mapping. We look at the letter that is before this C, which is A, right, and do last first mapping, so on, right. We can repeat this process, right, throughout these columns, right, first column, last column, until we reach the dollar sign here, right. That would be the end, okay. And this is the reference sequence that we generated, right. So, hopefully it is clear, right, how we can utilize this information of BWT and the first column using LF mapping to generate the original reference sequence. Okay. So, it means this is a very efficient way of storing the genome information, right. Just storing the BWT can give you the reference genome sequence, it can help in read mapping. Okay. So, we will see how we can utilize this information and last first mapping property to map reads in the next class. Okay. So, this is the this will be for the next class, right. We will utilize this BWT information and LF mapping to map reads to the reference sequence. Okay. These are the references that we have used. So, what we have seen is that we can generate this Burrows-Wheeler transform through string rotations and lexicographical sorting. We have talked about 3 steps, right, starting from the reference sequence, right. One is addition of this dollar sign, and dollar is lexicographically smaller than all the letters in the alphabet. And then we do the string rotations, right. We generate all possible rotations until we reach the original string, right. We come back to the original string, right. And in the third step, we combine the original string with all the rotations to do something called lexicographical sorting, right. And we have seen the process of lexicographical sorting and how we make those decisions for sorting, right. If the first letter is same, we look at the second letter, if second letter is same, we look at the third letter, and so on, right. And then we sort these strings and we generate this sorted matrix. Now, once we have generated this sorted matrix, what we have seen is that the Burrows-Wheeler transform, it takes same space as the genome, right. So, it is a very efficient way to store the genome data, right. It is not taking a huge amount of space like the hash table or the suffix array or suffix tree, right. So, that is good news, right. We can probably run probably in the desktop. We will see in the next class, right, whether that is possible, how much we will quantify the time and the memory requirement for read mapping. Okay. And what we have seen, right, this Burrows-Wheeler transform, it actually has very interesting properties, right. So, one is we can generate first column of the matrix from BWT, right. So, if you take the BWT, you can simply sort lexicographically, right, and that will give the first column, right. And we have seen a property called last first mapping, right, and utilizing this BWT with the first column and the LF mapping, right, we can generate the reference sequence. Okay. So, we will keep this in mind, and in the next class we will see how we can utilize this information, this BWT, the LF mapping, etc., to actually map reads against reference sequence in an efficient manner. And we will also look at whether we can handle reads that contain mismatches or sequencing errors, right. That is a very important requirement for us, and we will also see how we can how this method will deal with repeat mappings, right. So, multiple mappings of a read which will happen in case of reads mapping to repeat sequences in the genome. This is also a very important requirement, right. So, because many human genome or other big genomes, they contain many repetitive elements, right. So, there are many repeat regions, and you will get a lot of reads that will map to these repeat regions, and if this mapping reads to repeat regions takes a lot of time, that will actually increase the overall mapping time, right. So, these are the properties that we will need for a good mapping algorithm and to develop a good mapping tool. Okay. So, in the next class we will continue this discussion to see how we can map reads using this Burrows-Wheeler transformation and whether we can address those issues that we have seen earlier. That is it for today. Thank you.